Next is history of INSDC. INSDC didn't start as INSDC from its beginning. As far as early 80s, and in fact in 1979, first activity of INSDC began. At first, 35 researchers mainly from United States and some from Europe agreed on creating a united database to cover all kinds of sequences. Because at this time, DNA sequence method or Sanger method was developed and huge genome information started to cover journal pages. But those printed genomes do nothing for researchers because we could not search or locate subsequences from printed pages. So computer was necessary to search and compare obtained sequences. 3 years after this initial meeting, EMBL or EMBL Bank was started in Europe first. And right after, in the same year, 1982, GenBank was started at Los Alamos Science Laboratory. And these two databases were made publicly free and without restriction because both databases were run by public money and both databases were needed for all researchers working on sequences. So the very beginning of free and unrestricted access started almost 40 years ago. But soon after, curators or researchers working on these databases realized that more workload is needed because there are journals published not only in Europe or in the United States, but many papers were published in Japan or in Asian region. And in Japan, many papers were published in Japanese. So there was a demand that those sequences written in Japanese journals be covered. And DDBJ joined the activity in 1987. So initially, each database worked on different journals, but very soon we started to exchange our results. And at that time, most data were manually input and exchanged using magnetic tapes. So the update frequency was much slower, but we started the fair and unrestricted exchange of information from the very beginning for this reason. If you want to know more about why Japan joined this activity, please refer to the reference at the bottom by Stevens, Globalizing Genomics, the origin of the INSDC. This collaboration was culminated at the time of Human Genome Project started in 1990. The Human Genome Project, or HGP, started by NIH and DOE in the United States and collaborated by Wellcome Trust in the UK. And in Japan, Japanese government participated to this international project. At that time, the initial leader was James Watson. You see his face at the right corner of this slide. And under this international collaboration, GenBank was relocated from Los Alamos Science Laboratory to NCBI in Bethesda, Maryland, to the very close place to the Washington, D.C. headquarters. And EBI was newly established in Hinxton, UK, using much funding from Wellcome Trust. So the current INSDC was formed early in 1990s. And from the 90s, we started daily exchange of our update records. From 1996 to 1998, in three consecutive research agreements, all researchers confirmed the immediate publishment of human genomes, mainly because of the very competitive and severe race against the Celera Genomics, a commercial company who claimed to publish all human genes before the International Human Genome Project. And through the severe competition, International Consortium declared that we will publish all sequences immediately, even before paper publication or even before research analysis. This is called now Bermuda Agreement. And after Human Genome Project ends, there was another important agreement in Fort Lauderdale. And in this agreement, not only researchers, but also journal publishers and also research funding agencies agreed on not immediate, but publication, free and unrestricted publication of all genomes from major sequencing centers. So from these particular agreements, we now see that all major genomes are publicized for free without any restriction, basically from ISDC. And at the beginning of next generation sequences, EmbryBank was renamed formally to European Nucleotide Archive, or ENA. So now ISDC is said to be formed by DDBJ, ENA, GenBank, and these three institutes cover all primary sequences for all life sciences. Here we call primary repository because we receive all data for all scientific publication from everybody fairly, and we archive all records permanently without any change. So primary repository is very important for research integrity. If you want to reproduce or double check any result, you need to come back to ISDC and check the original record. For this very purpose, we need to keep our open access with minimum access restriction. Of course, for personal genomes, we need to put some restriction, but we try to make this restriction to the minimum. And our principle is compatible with what is called FAIR principle. FAIR, each letter stands for findable, accessible, interoperable, and reusable data principle. But very often, this FAIR principle is referred to in conjunction with CARE principle. C for collective benefit, A for authority for control, and R for responsibility, and E for data ethics. I personally agree with the CARE principle, but the policy of ISDC is incompatible with the second part, authority for control, because we cannot give the data submitter any authority to control data, for example, deleting data. That's why we always say that FAIR principle is okay, but CARE principle is somewhat incompatible. Well, for the last part, I will briefly describe our institution, National Institute of Genetics, to which DDBJ belongs. National Institute of Genetics is located in Mishima City, Shizuoka, and Mishima City is 100 kilometers west of Tokyo. At NIG, we have about 30 research laboratories, and DDBJ is part of them. At DDBJ, we provide not only databases, but we have supercomputing facility to maintain, operate, and maintain DDBJ, and also for researchers to use our computing facility. NIG also has Genome Sequence Center and Bioresource Center, or National Bioresource Project called NBRP. This NIG supercomputer will be explained in the next lecture by Dr. Okuda, and it is basically available for only Japanese researchers. But some participants have already obtained permission to enter or log into NIG supercomputer. This is possible on condition that you, foreign researchers, collaborate with Japanese researchers, and on condition that those collaborating Japanese researchers take 100 responsibility as a guarantor. So with some special condition, our computing system is available for foreigners too. And the number of current NIG computer user exceeds more than 1,700 per year. So it's a very crowded system, but you can use this facility for free. This is a very unique facility for all researchers in Japan. And at NIG, we provide also various opportunities, not only for Japanese students, but also for foreign students. For example, NIG serves as a department of genetics at Sokendai University, but we also provide various courses for postdoctoral training and also for using our shared facilities. So if you want to study bioinformatics or if you want to study genomics, please check the website of National Institute of Genetics. We provide international collaboration funding, and also we provide opportunities for students to study here.